Okay, so let's get started. So welcome everyone to our today's Zoom seminar. Particularly, I want to thank Gaofeng and every audience spend the weekend with us. So today it's really my pleasure to welcome our speaker, Dr. Gaofeng Fan from ShanghaiTech University. So Gaofeng currently is an associate professor at the School of Life Science and Technology of ShanghaiTech University. So Gaofeng completed his undergrad study in biology from Department of Intensive Institution, Nanjing University. And then afterwards, he pursued his PhD training in biochemistry from Rutgers University in New Jersey, US. And then after he completed his PhD work, he pursued his postdoc training in Cold Spring Harbor Laboratory. And then since 2017, he joined ShanghaiTech University, start his independent research program. So Gaofeng's research actually mainly focus in the signaling, transduction, and metabolic regulation of T cells, with particular interest of how to better assistant in cancer immunotherapies through understanding these biological processes. The main focus of his research program include to study the role of metabolic molecule NAD and its regulatory pathway in tumor immunity, focusing on the biological function of NAD metabolic enzyme and the channel protein that NAD utilized. And second part of his research focus is to understand the role of tyrosine phosphatase, PTPN6 and SHIP1 in immune cells. And last but not least, his group tried to studying the role and the molecular machinery of tyrosine kinases and then phosphatase in the spread, metastasis, and drug resistance of ovarian cancer. So which I think he's probably going to tell us a lot today about how the T cell transduction in cancer biology. With different all the achievement during his postdoc training, and then after his independent research program, he received numerous awards, which include the Shanghai Tech University Award for Excellence in Teaching, both in 2017, 2021, 2022. And then Eastern Scholar, Distinguished Professor from 2019, and then follow up the funding support, and then Pujiang Talent Program on 2018. So I think without further ado, Gaofeng, thank you very much to join us for today's talk, and then we're looking forward to your talk.
Thank you so much, Chuan, for your kind introduction. And it's my great honor to include me in this seminar series to share our recent work. I have been attended this series seminar for many times, and I really benefits from all the kind of inspiring ideas and also the very exciting results from all the colleagues worldwide. So today I will mainly focus on the signal transduction and metabolic regulation in T cells. So I don't think I have to further repeat my personal resume. So my lab is mainly focused on the signal transduction, and I have already established my lab in Shanghai Tech University almost 7 years. And and today I just share two stories basically completed during these 7 years, and share some thoughts on signal transduction and immunotherapy. So it's been almost 50 years since the Nixon has signed a bill conquer the cancer, and these days, from the 17th, I think the front line for the cancer patient treatment is basically surgery, and still it's a very productive treatment regimen for today's patients. And then they develop the radiation chemotherapies and for kind of the targeted therapies. And then back to 2000, at the end of the 1990s and the early of the 2000, the first molecular targeted therapy targeting the tyrosine kinase BCR-ABL has been developed, and Gleevec has been developed by the Novartis companies. And this opens the era for the molecular targeted therapies. And for the recent decade, I think everybody has been inspired and encouraged by the immunotherapy, featured by the ICB and Doppler transfer to mainstream therapies worldwide, which can benefit lots of patients. Because of this excellent performance in the clinical end, so the science has listed the cancer immunotherapy as a breakthrough of the year in the 2013. I think in this audience, I don't have to spend a lot of time to introducing the both therapies. One is the immune checkpoint blockade ICB therapy, and another is CAR-T therapies. The main ideas here is there is one molecule called PD-L1, which has been expressed on the tumor cells, and its cognate ligand, the PD-1, expressed in the T cells. Through this ligand and the receptor ligation, the tumor cells can inactivate the function of the T cells, which can help them to escape from the immune surveillance. According to these breakthrough discoveries, scientists have developed the antibody which can interfere with these co-inhibitory ligations. And these antibodies immediately make a big impact both academically as well as clinically. So the scientists got lots of these findings helps a lot of the treatment revolution in the clinical end. However, during the course of the drug development and also the patient treatment, several major challenges has been also occurs. As I list here, some patients are not respond to PD-1, PD-L1, and even patients can respond to this treatment. The only 20 to 30% of patients can benefit from the ICB. And also, another big issue is because of the immunosuppressive microenvironments, this ICB method didn't make a bigger impact on some solid tumors. And also for the CAR-T therapies, I think this therapy is also very revolutionary developed, and it hijacks the ideas or all the ideas from synthetic biology. So the scientists took the antibody design and combined with the activation domain of the TCR zeta chains and make a chimeric protein called the CAR proteins, and transferred to the T cells. And based on these ideas, they developed the first generation, second generation, and the third generation. So basically, the core strategy is very similar, only with difference between some like putting the different co-stimulatory domain like CD28, 4-1BB, or combined together. And again, these therapies also make a big impact to the therapeutic end. However, it also has some issues during the course of this treatment. And again, like for example, the immunosuppressive microenvironments also make a big challenge to this method. So when I established my lab, one key questions we are very interested to address is, can we identify the factors that are the key determinants for the function of T cells? As long as we can understand the mechanism for the T cell activation, especially within the immune suppressive environment, for example, from the signal transduction end, or maybe from the metabolic end, maybe we can better shape the regimen for the immunotherapy treatment. So with this idea in mind, I split my initial lab into two groups. One is mainly focused on the signal transduction and trying to find the important signaling modules, which is important for T cell signaling output. And maybe later we find it's also important for the T cell fate determination. And the second part, we are mainly focused on the metabolic regulation and trying to find the metabolic nodules, which are also important for the T cell activation and the function maintenance. So today I want to share two small stories and to shed some lights what we are thinking right now in the lab. The first part is basically related to the CMIS and the PTP-N6. I think Dr. Wu, your colleague, Paul Love, is also works on the CMIS and also is one of his favorite molecules. So, and I just want to share some thoughts from my lab on these molecules. So, I was trained as a biochemist, basically within the tyrosine phosphorylation field. So as we all know, back to 1979, Tony Hunter and Ray Erickson has identified the first tyrosine kinase called SRC. It's also the first oncogene has been identified in the human genome. And since then, 19 tyrosine kinase has been identified. The tyrosine kinase has ability to transfer the phosphate from the ATP to the hydroxy group from the tyrosine residues. And this process is wholly reversible. And my postdoc mentor, Nick Tonks, was the first one to identify the reversible enzyme called the tyrosine phosphatase, which can remove the tyrosine residues from the phosphorylated tyrosine. It was discovered in 1987. It called the PTP1B. Since then, 105 tyrosine phosphatase has been identified. And another group of protein within the human genome called the SH2 domain containing proteins, which can recognize the phosphotyrosine motif and performs the signaling cascade and to complete the whole circle of regulation for the tyrosine signaling, phosphotyrosine signalings. And Hannah Fusa and Tony Paulson are the leading scientists who are in charge of identifying the SH2 proteins. In terms of the druggable targets and FDA-approved drugs, tyrosine protein kinase are among the top as a major protein family for drug development, and 50%, 56% for the druggable targets, and 30% for the FDA-approved drugs, just 3% behind the GPCR. And not only for the kinase, tyrosine phosphatase has also gained lots of attention these days. Again, Novartis, back to 2016, published a Nature paper and disclosure a molecular glue-like inhibitor called SHP099. This molecule can inactivate the tyrosine phosphatase called SHP2. The another family member which I'm talking about today's talk is called SHP1. They are very close relatives, and these molecular glue inhibitors can lock the SHP2 in closed conformation and inhibits its oncogenic functions. And since the discovery of this SHP099 and as well as its improved version TNO155, Novartis invests lots of money and also lists as one best at first-in-class aspiration from their 2019 annual reports. So phosphatase starts shedding their stigma of undruggability since then. And I would either further to emphasize, when we look deeply into the ICD treatments, actually, by the ligation of the PDL1 and the PD1, the function of this co-inhibitory signaling have to focus on the activation of the phosphatase activity in the end. So anything, if we can find anything interesting can targeting the phosphatase activity, will clearly benefits the immunotherapy. This is also the one strong support for the ideas of our labs and keep us moving forward. So, but I was trained as a biochemist, and whenever I propose idea to my former boss, Nick Tonks, talking about like we have to antagonize the function of some enzymes, particularly for the tyrosine phosphatase, he was always very careful and said, if you want to inhibit any enzyme, particularly like the tyrosine phosphatase in your systems, you'd better have a big picture or maybe clearly definition for the function of this tyrosine phosphatase within particular environment. For example, like here, immune systems. So I have a desire, or maybe I have some obligation to providing all the potential function about the any phosphatase I want to inhibit. So since the starting of my during the starting of my lab, I don't have that kind of resource to do everything at the same time. So I can only simplify the question like, do we have any ideas to providing the potential substrate pool for any phosphatase we are trying to looking at? If we have this kind of the ideas, or if we have this kind of the data, maybe we can make some more careful judgment. What's the pro and what's the cons when we talking about the inhibition any tyrosine phosphatase we want to. So I just give our simple answer. Can we develop a robust and systematic approach to identify substrate for protein tyrosine phosphatase, particularly here PTPN6 in immune systems? So actually we are very lucky. Nick, back to 25 years ago, and has been identified the whole mechanism for the tyrosine dephosphorylation. According to this dephosphorylation mechanism, he also developed a substrate called a mutants called the substrate trapping mutants, which can help us to better shape the way to develop some like systematic approach to providing substrate. So here is a classical substrate for the tyrosine phosphatase, labeled in red, and the three key residues labeled here as white as are the active sites for the tyrosine phosphatase. As for the active sites for tyrosine phosphatase, so the key residue is called the cysteine. The cysteine can make a nucleophilic attack to the phosphate, and this will make the formation of the bond S and the P, and makes the PO bond to break up. And this oxygen will grab the proton from the side chain from the aspartic acid and finish the dephosphorylation. And this aspartic acid is conserved in the whole PTP families. And the next thing, and based on these findings, Nick proposed, if we mutate this aspartic acid to the alanine, which will make the release of the proton more difficult and also shorten the side chain, it will keep the phosphate group attached to the cysteine and makes the substrate more sticky to the mutant enzymes. they make this strategy called the substrate trapping strategies, and it will help to create some time windows for biochemists to fishing out the substrate. So based on this technology, and everybody in the PTP field, when they talking about the substrate identification, they instantly make the mutation, the DA mutation to the phosphatase, and purify the mutant proteins, and incubate this purified protein with phosphorylation, tyrosine phosphorylation enriched lysates, and do the IP, cut the band, and send to the mass spec for the identity identification. However, this method first is quite laborious, and second, some loosely bound interactions will dissociate when they go through all the hard wash biochemical buffers. So it makes a lot of false negative results through runs of this biochemical experiment. After I joined ShanghaiTech universities, so my colleague Zhuang Ming developed a method called the puppet. It's a type of the proximity labeling strategies. Basically, she cloned a E3-like protein called the PUF-A from the bacteria, and also a ubiquitin-like protein called PUP-E, also from the bacteria, and put these systems back into the mammalian systems. And any interesting protein, we can make a fusion protein with PUF-A, and any binding proteins close to our protein of interest will be labeled with PUP-E. And we can do some biochemical modification on the PUP-E, like the biotin labeling. So all the labeled free proteins can be enriched by the streptavidin beads. And then we can perform the mass spec to systematically to providing the whole spectrum of the substrate. So the rest will be very straightforward. We combine the substrate trapping system along with the puppet tagging systems. And basically, we are focused on the wild type and the substrate trapping mutants of the phosphatase. And anything more sticky to the trapping mutants, we will be considered as a potential substrate for our tyrosine phosphatase. We call it as the Pepsi puppet enhanced PTP substrate identification systems. So we applied this system to our favorite phosphatase called the PTP N6 SHIP1. And we performed these systems in the jerk T cell tumor cell lines. And as you can see here, we identify the thymus as the top hits through the 3 rounds of the experiment. And biochemically, we also confirmed that the trapping mutants show more robust binding affinity with thymus, both in the 293 and jerk T cell system. So thymus stands for the thymocytes expressed molecule involved in selections. And back to 2009, 5 groups has identified at the same time that this molecule play a very important role during the T cell development. As they developed several distinct methods to genetically disrupt the expression of the thymus. And what they found is the T cells, a lot of T cells are development of the T cells stopped at the double positive stage, and they never go through to seldomly go through to the single positive the stage and for the further maturation. So first thing first, so our mass spec data suggests the thymus potentially as a potential substrate for our phosphatase SHIP1 PTP-N6. So the first thing we want to identify is thymus can be phosphorylated during the T cell activation, or maybe during the upon any important kinase during the TCR activation can phosphorylate stimulus. So there are several. We test the whole spectrum of the major tyrosine kinase downstream of TCR, and we found that only LCK, but not the ZEP70, can phosphorylate stimulus very robustly. And this phosphorylation, we also did perform the phenylalanine scanning, and we found that this phosphorylation mainly happens at the tyrosine 34 residues. And so based on this finding, we further perform the in vitro kinase assays with stimulus wild type and stimulus Y34 as substrates. As you can appreciate here, the wild type stimulus can be phosphorylated by LCK, but the Y34 mutation totally abolish this tyrosine phosphorylation. We also performed the mass spec identification and to confirm that we can find the PY34 peptide fly in the mass spec machine. And this is based both in the purified stimulus protein as well as immune precipitated stimulus proteins from 293T cells. And since these tyrosine residues are conserved among species, so we instinctively to make the antibodies against these phosphotyrosine residues. At the time, my lab is not that rich. No, we don't have enough funding. So we only make a polyclonal antibody to against this site. And turns out the whole antibody production journey is quite pleasant. And this antibody can recognize particularly the phosphotyrosine motif for the stimulus. And when we mutate this tyrosine, it totally lose tyrosine phosphorylation. My students also replaced all 18 tyrosine to phenylalanine, but only keep the 34 tyrosine on the stimulus. And this protein can still be expressed and can still be phosphorylated by the LCK and can be detected by this antibody. So the antibody is quite good. So with this antibody in hand, so we can ask the next questions. Does SHIP1 can dephosphorylate stimulus at this particular site? We perform both in vitro dot plots as well as in cell overexpression system, and both systems confirm that the SHIP1 can dephosphorylation stimulus at tyrosine 34. And here is some small trick for the tyrosine field. So, and this is also common for the SHIP1 and SHIP2. So SHIP1 and SHIP2, they are balanced between the active state and the inactive states. And there is one classical mutation called the EA mutation at their SH2 domains can totally make it constitutive active. So we, in the later part of the studies, we use these constitutive active phosphatase active mutants for our experiments. And you can see here it can actively dephosphorylation this peptide. And so we see the dephosphorylation, and another thing we see is that when we have this tyrosine phosphorylated at the 34, the trapping mutants, the DA mutants, can make a strong binding with this substrate. However, if we mutate this site to the phenylalanine, we totally lose the phosphorylation ability, and we also totally lose the trapping ability for the trapping mutants. So both dephosphorylation and the trapping mutants confirm that tyrosine 34 of the stimulus is a bona fide substrate for the SHIP1. And what about in the endogenous level? So we also took out the LCK kinase. We totally lose the phosphorylation signals. When we took out the SHIP1, the phosphatase, we boost the signal. And as you can see here, the signal can be boosted but not totally abolished. So when we took out both SHIP1 and the SHIP2, we totally lose this tyrosine phosphorylation. These tyrosine phosphorylation signals can last forever. suggesting SHP1 and SHP2 are both in charge of the dephosphorylation of these residues. So far we know that the LCK and the SHP1, probably SHP2, play the opposite function in regulating the CMIS phosphorylation. And the CMIS play a very important role during the T cell development. The next question we want to address, does this phosphorylation event also play a big role in T cell development? So we try to address these questions. We collaborate with Zhuo Aowei, and he helps us to make the transgenic mice for the Y34 knocking mice there with CRISPR technologies. And during the course of making these knocking mice, we also have some autoframe shift on these genes. So we also got the total knockouts of the CMIS strings. So later we can compare the different phenotype between these three strings for the biological research. So, and importantly, this knocking mutation didn't affect the expression and the stability of the CMIS proteins. And we look at the T cell development for these different strains. And first, we confirmed that when we took out the CMIS genes, we also repeated data which has been published by 5 groups. We do see the significant loss of the CD4 T cells as well as CD8 T cells. And also, we found that the Y34F, the phosphorylation deficient mutant mice, also showed, although mild, but also significant reduction of the CD4 single positive cells, as well as CD8 single positive T cells. After the careful the calculation and also the counting of the many mouse, we found that the CD4 defects is more significant is significant when we in our knocking mice. CD8 T cells are not significantly affected. I will come back to this point in the later talk. Due to the time limits, so I just summarize the major phenotype on these slides and move to the mechanistic studies. First, when the phosphorylation deficient mice show a significant reduce the CD4 single positive thymocytes in these knocking mice, and the CD8 single positive thymocytes are largely unaffected. And in peripheral, the number of CD4 T cells, but not CD8 T cells, is remarkably reduced in CMIS knocking mice. And also the IL-7 induced homeostasis of CD8 T cells is not affected in the knocking mice. So what is the molecular mechanism behind all this regulation? So right now we have 3 genotypes of the mice. What we can do is purify the thymocytes, and further we can purify the phosphatase in these 3 genotypes and testing the phosphatase activities in vitro. So these slides are quite important. So we got 3 interesting findings. First, so upon activation of the TCR, not only kinase but phosphatase can also be activity of the phosphatase can also be enhanced. And this enhancement can be totally abolished by the disruption of the CMIS. And most importantly, this phosphatase activity elevation can be totally abolished by the point mutation at Y34, suggesting these tyrosine residues upon phosphorylation is important for the phosphatase activation. So with this key data in mind, so we synthesize this peptide with tyrosine phosphorylated and unphosphorylated. And we also found that this particularly PY34 CMIS peptide as listed here, can elevate the phosphatase activity of the SHP1. And this sequence of this peptide is also unique. So when we use the PY132 lead peptide, it cannot be used as a phosphatase agonist. And also the Y34 semis peptide upon unphosphorylation is also cannot used as a phosphatase activator. And also when we make a Y34D mutation on this peptide, it can also to mimic the phosphorylation, it can also enhance the phosphatase activities. So both data suggesting this tyrosine phosphorylation is important to activate the SHP1's activity. So to save time, I just first present our working model first and show more supporting data to support this model. So basically, the SHP1 has two conformation. One is closed conformation and one is open conformation. And these two conformation they have the transition mechanism to balance between these two conformation. And upon activation, the CSH2 domain will lift up and switch for like 110 degrees. So the NSH2 domain will switch back to the PTP domain, and this rotation will expose the PTP active sites and make the phosphatase more active. And here, our findings suggest the Y34 upon phosphorylation by the semis LCK, it can bind to the PTP domains and will fix this PTP in an active conformation. So keep the phosphatase activity alive. So, and when we look at the alpha fold, we can see that the tyrosine 34 is around on the surface of the PTP N6, suggesting this kind of the residue is easily be modified by the kinase and the phosphatase. So we further did two more experiments to support this model. First is the SPR experiment. We measure the binding affinity between this peptide with both the SH2 domain as well as the PTP domain. We do find that the PY34 semis and as well as Y34D semis peptide shows more significant binding affinity with the PTP domain rather than SH2 domains. We use the SH2 domain binder, the PY6A6PCAM peptide as our negative control. And furthermore, we further look at the PY34 semis, the peptide, in which place they can bind to the PTP N6. Through the HDX analysis, the hydrogen deuterium exchange mass spec experiment, we mapped a region upper 9 helix on the PTP domains, which can bind to the PY34 peptide. And interestingly, this region has 3 lysine residues of the region. So when we mutate these 3 lysine residues, we totally abolish this regulation, suggesting maybe these positive or maybe negative, positive and negative charge interactions helps the binding of these 2 molecules. So here is the final summary for this part of the talk. So as we all know that during the TCR development, the CD4, CD8 double negative cells were first developed to the CD4, CD8 double positive. And then they will move to the CD4 positive and the CD8 negative stage and make a thinking which path they want to go, either the CD4 single positive or maybe or the CD8 single positive. And here we also have important signaling switch. So for the CD4 development, they using TCR signals consistently, but for the CD8 development, they switch from the TCR activation to the interleukin signaling for the further development. So the CMIS play both a tyrosine phosphorylation dependent and independent role during the TCR development. And it can only be phosphorylated by the TCR signaling, but not the IL-7 signaling. That's why the CMIS phosphorylation deficient mutants have the more profound defects for the CD4 TCR development, but not for the CD8 TCR development. This data I don't have time to share here, but if you're interested, you can look at our paper just come out this year. So I'll make a quick summary for this part. So we developed a method called the PEPC, which can help to the substrate identification. And we use this platform to identify a new substrate for the SHP1 called the CMIS. And the CMIS is oppositely regulated by the kinase LCK and the SHP1. And this tyrosine phosphorylation is important for the SHP1 activation. And CMIS plays both the phosphorylation dependent and independent role in regulating TCR development. So currently, we also noticed that the CMIS, the expression of the CMIS is quite unique to the TCRs and also quite unique to the lineage of the TCR development. They only express at the double positive stages, and then they quickly goes down during the single positive stage. So two projects ongoing right now. We are very interested in deciphering the transcription regulation as well as the protein quality control of the CMIS. So hopefully next time we can give you another update on this particular molecule. So I will switch my gear a little bit at talking about some metabolic thinking in TCR activation during the progression of the project in the lab, and mainly focusing on the NAD. And I will tell you why we are particularly fascinated on these metabolites. So as I said at the beginning, the key question is we want to identify the signaling module as well as metabolic modules, which is important for TCR activation. And so we collaborate with Gaofeng and to do the genetic screening. Gaofeng Wang, my colleague in ShanghaiTech, to do a genetic screening to identify all key genes involved in TCR activation and inactivations. At the same time, Alex Marston also performed a similar screening. And the only difference is we performed in direct T cells and the Alex Marston performed in primary T cells. So with both data sets on hand, we overlay both data sets and to see and to highlight the top 50 genes which are common in the both studies. At that time, we don't have any clues which genes we may want to follow. So after talking with all the collaborators, so we decided to perform a second screening. And these screenings were mainly focused on the signaling nodule inhibitors as well as the important metabolic enzyme inhibitor. We performed a pharmacological screening with all the small molecular inhibitor library. So we bought a compound. We bought a library from Stylag, which contains 199 compounds. And interestingly, we perform when we look at the data from the GRCAT human T cell CD8 T cells, and we found 3 inhibitors which targeting the same genes called NAMPT are also jumped out as the top hits. And this gene has also been listed as the top 50 hits in the first genetic screening. The NAMPT stands for the nicotinamide phosphorybtransferase. So what this gene do during our mammalian system, in the cells? It turns out it's a rate-limiting enzyme for the NAD biosynthesis in the mammalian systems. So basically, there is 3 pathways in cells to produce NAD. First is called the Prince-Handler pathway, starting with the substrate called the nicotinic acid. And the second pathway is called the de novo biosynthesis pathway, starting with an essential amino acid called tryptophan. And it goes through almost 8 steps to finally produce NAD. And the shortest and the most convenient way to produce NAD is through the salvage pathway. And the NAMPT is in charge for the NAD production of this pathway. With only 2 steps, it can convert the nicotinamide to NAD. So first thing first, so we want to make sure that our screening results is correct. So we set up 4 conditions for our experiment. One is control group, one is NAD plus group, and one is the inhibitor, NAMPT inhibitor FK866 groups, and one is the inhibitor plus NAD rescue groups. And we use the T-cell activation marker as our readout. As you can appreciate, upon treatment of the NAMPT inhibitors, the T-cell activation are dramatically reduced. However, this phenotype can be totally rescued by the re-addition of the NAD, suggesting that the NAD is quite important for the T-cell function maintenance and also the activation. And further, we look at the phosphor ERK and also the general TCR signalings, and we got the, and as well as the calcium flux, we got a very similar result. Upon the NAMPT inhibitor treatment, we totally abolished the tyrosine phosphorylation signaling upon the activation of TCR, and this reactivation can be rescued by the re-addition of the exogenous NAD. And so what about in the physiological relevant conditions? We took 18 ovarian cancer patient samples from the hospitals and compares the NAD level between the T cell in PBMC and NAD level from the tumor infiltrated T cells. As you can see here, the TIRS has significantly low NAD compared to the PBMC, and this result has been repeated in the mouse model, the 8 mouse melanoma models in-house. So, as we all know, this picture I took from the undergraduate textbook. NAD and NADH acts as an electrocarrier in the oxidative phosphorylation process, and it, especially for the NADH, can be function as a substrate for the electron transportation chain, and finally to help the production of the ATP. And also, NAD can be a co-substrate for the PARP and the SIRT enzyme to perform the ADP ribosylation as well as the deacetylation. So we test all the possibility from the oxidative function as well as the co-substrate functions with inhibitors. And turns out the function as a co-substrate is not quite significant during the T-cell activation. So basically, we are still keep focusing on its function as an electrocarrier during the ETC transported chain and ATP production. So we perform the metabolic mass spec and compares the metabolic change during the FK866 treatment. As you can appreciate, upon the NAMPT inhibitor treatment, all the metabolic pathway, major metabolic pathway like the TCA cycle, glycolysis, ETC, has been significantly changed. And when we look at the ADP, ATP, and AMP levels, as you can appreciate, upon FK treatment, we see a dramatic loss of ADP and ATP, and reversely increase of the AMP. And this can be this phenotype can be totally rescued by the NAD re-addition. So suggesting that the change of the NAD may be directly linked to the ADP production, ADP status of the T cells. So we further collaborate with Wang Ruoning in Ohio State Universities using the isotropic labeling experiment to evaluate the function of the glycolysis and the glutamine lysis upon treatment of the NAD inhibitors. And as you can appreciate here, both glycolysis as well as glutamine lysis has been dramatically decreased upon the FK treatment. Since glutamine lysis, this process using the TCA cycle, and this happened in the mitochondria. So we go forward and look at the morphology as well as the function of the mitochondria upon this treatment. As you can appreciate here, so upon the NAMPT inhibitor treatment, we see the violence of the CRISD are dramatically decreased, as well as all the mitochondria, the morphology of the mitochondria are dramatically changed. And the re-addition of the NAD can totally rescue this phenotype. We use the seahorse to measure the OCAR rates during these 4 conditions. And the inhibitors can totally flatten the curve of the OCAR usage, and this decrease can be rescued by the NAD re-addition. So looks like that the NAD plays an important role for the T cell activation. So next, we're trying to further to evaluate the in vitro activity as well in vivo activity of the T cells during these 4 conditions. So as you can here, we use CAR T models, the CD19 CAR with to illustrate the function of this the importance of the NAD regulation. As you can appreciate here, upon the inhibitor treatment, it dramatically decreases the toxicity of the CAR T cells during the in vitro killing experiment. And when we look at the key contributor like the granzyme B, interferon gamma, and the IL-2 production, it also have the very similar shape. The FK treatment can dramatically decrease the production of these key effectors, and the NAD addition can rescue this phenotype. And finally, so we want to testing this kind of the findings in vivo. So we use a CAR T model and as well as the ICB models to testing these functions. As you can see here, the CAR T cell already show a very dramatic effects on the T cell clearance. However, with the addition of the NAD as well as its precursors, the combination treatment can even boost the tumor cell clearance during the experiment and prolong the lifespan for the mice. So due to the time limits, so I just give you our working model here. So basically, the NAMPT mediated NAD biosynthesis is crucial for the activation of the T cells. And NAD regulates cellular energy metabolism by influencing the glycolysis and the TCA pathway in T cells. Supplementation NAD can significantly enhance the efficacy of cancer immunotherapy. So I only show the CAR T data here. So actually, we also did the PD-1 as well as the CTLA-4 treatment. And the PD-L1 and the PD-1 treatment also give a very significant efficacy when do the combination. So we published the paper 3 years back, and in Cell Reports. If interested, you can look at the paper. Basically, we also identified that the MYC as well as collaborates with the CARB can transcribe the NAMPT activation for the transcription activation. So for support the NAMPT production in the T cells. So which I don't have time to show here. So during the course of this project, actually, the Weiping Zhou from the University of Michigan have very similar ideas. So talking about the metabolic competition between the tumor cell as well as the immune cells. What they found is that the methionine metabolism is also important for the immunotherapy. Basically, the tumor cell can upregulate the methionine transporter, or called SLC43A2, and compete with the T cell for the methionine uptake. Without methionine, the T cell can no longer properly express the STAT5, which supports the survival of the T cells. So this since these two papers, I think many very similar studies explosively published, talking about all kinds of metabolites, the significance of metabolites in the immunotherapy. So make a quick summary for the whole part of my talk. And first, the NAMPT mediated NAD biosynthesis is crucial for the activation of the T cells. And well, I don't have time to show you here. My lab also found that not only T cells need the NAD, for the immune cells, for the neuron cells, they also need NAD for their functions. So for the tumor cells, they also need NAD for their survival. So if we can develop any strategy to targeting the NAD metabolism in tumors, we can effectively kill the tumor cells as well as in the in vivo models. So basically, our lab took the PROTAC strategies to degrade the NAD synthesis enzyme NAMPT and show a very encouraging efficacy in the tumor cell clearance, both in vitro and in vivo. And also, we don't have time to share with you at today's talk. My lab is also working on the plasma membrane transporter identification for the NAD. So all the re-addition with NAD can totally rescue the phenotype in the T cell functions. Since then, we think maybe the NAD transporter is also exist on the plasma membrane, which can using as a safeguard for the T cells to uptake exogenous NAD from the environment. So I quickly give some like the screening strategies here we used in this work. Basically, so we think the NAD, they have the cells have two ways to maintain NAD level in cells. One is from the internal biosynthesis. One is from the outside uptake. So if we block the internal NAD biosynthesis through the NAMPT inhibitors, so the cell needs to uptake NAD from outside to keep cell alive. And on this, so in this scenario, if we also knock out the genes one by one, and if we knock out the right gene for the NAD uptake, so the cells can no longer keep the NAD concentration, so cells are going to die. So with this life and death screening strategy combined with RNA-seq, we kind of pinpoint the NAD transporter on the cell membranes, and hopefully we can update this story in the next seminars. So for the future plan for the NAD parts, so basically right now we are my lab is actively involved in the NAMPT PROTAC the improvement and development, and this is aiming for the pan-cancer treatment. So, and because each cancers they have the different gene expression patterns, but they all need NAD for the survival. So we propose this kind of metabolic targeting strategy maybe can targeting the multiple cancers for the treatment. And for the immune cells as well as for the neuron cells, we are kind of developed two strategies to enhance the NAD replenishment. One is from the exogenous NAD replenishment by engineering the NAD transporter. The another way is we trying to develop some like NAMPT activators to enhance the endogenous NAD biosynthesis. So with these two directions, so maybe we can finally to address some question like the immunotherapy boosting, axonal degeneration delay, as well as the anti-aging experiment. So finally, I want to thank the people who are working, performing all this science. The Jia Li and Zheng Zhou here. Jia Li, Zheng Zhou performed the CMIS PTP N6 stories. Yue Tong performed the NAD stories, and Yu Feng performed the NAD transporter stories. Zhu Xiaotong, Li Ye, Chen Li performed the NAMPT Protect stories. And I also want to thank all the collaborators inside the ShanghaiTech as well as outside the ShanghaiTech for their continuous support to my research. And finally, I want to thank all the fundings I received during the production of all these projects. Thanks for your attention. I will stop here and leave some time for the question. Thank you.